I would actually love for you to talk about that because when Strongest Hearts is out talking to our community and to our donors, we talk about how fast CRISPR is comparatively to small molecule and traditional pharma, right? And it's one thing to say it, but there's actual real world examples now, like sickle cell and some of these other things that are now FDA approved. Maybe just like for like a minute, like talk about that, because that's the path we're going down.
So let's start with like what the small molecule, because that's the world we're coming from, right? So on average, it would take 20 years and 2.4-ish billion dollars
with a B
to get from a bunch of chemicals to something that is an approved drug on the market.
that also won't kill you.
Yeah, like aspirin and things like
Yeah.
that. And that's in the best case scenario if that thing actually exists. There's no reason that there's a chemical out there that we can take that won't kill us. It'll go to the right place. It'll actually have some effect.
Right.
It may not even exist. So we may be, and it's part of the reason why that number is really so high, is because it covers a lot of failures that are rolled into that.
Right.
Like all the opportunity cost of all the programs that didn't work out. So $2.4 billion for something that takes 20 years. So if we think about that 20 years, the first 10 years of that is trying to identify that compound. So going from like millions of things to one, and then the next is basically advancing that compound through cells and animals, large animals, and then into human trials, phase 1, phase 2, and phase 3, then approval. So that's 10 years. The beginning half is 10 years. If we now look at CRISPR, that, what would really have been 10 years, it's been collapsed to a couple of hours, in a day.
Wow. Because we know where to go. So and we know the methodologies and those sorts of things.
Exactly. So what happened in the early 2000s, we sequenced the human genome, and we've been refining that. So we know what essentially the reference is for a human. We know what the normal is. At the same time, people have been cataloging these mutations. So we know things, when this changes, we end up with this disease.
Right.
So we now know what right is and what working is and what broken is. And if we can go from one to the other, that's kind of the final loop in this, really this, type of scientific, it's really multiple scientific um, uh, areas of like study that are all intersecting at the same time.
Right. Like Human Genome Project, CRISPR.
Yeah.
And then adding to that, and this is something that I'm for my other life kind of excited about. Some technologies are also accelerating this, correct? I mean, we've talked about the PCR machine AI. Maybe you could talk a little bit about the technologies that are actually really helping to push this along at an even faster rate than ever before.
Absolutely. So the biggest thing I think everybody is hearing about is going to be the artificial intelligence, right? So there's no question that that is making huge impacts in how we do research, how quickly we can move things forward. So we were talking about, how long it takes to develop a drug. Well, now, we have one thing like, CRISPR that collapses basically decades of work, and also the chances of something being successful are way, way, way higher. So we've seen that with sickle cell. We've, with an approval that took basically 5 years instead of 20 years and, $2.4 billion. So the other thing that I think now is really making an impact is, as I mentioned, artificial intelligence. So that's something that we've been using. So particularly with DMD, one of the things that we do is you can imagine there's a lot of places in the genome. So how do you figure out where's the best place to go? So what we do is there's slightly different levels of artificial intelligence. Generally, like artificial intelligence is a broad like category. You then have something called machine learning, and then you have something called deep learning. Deep learning is what we've used on the genome and with the sites to figure out what are those best candidates? And what that allows us to do is really speed up something. So instead of us needing to look through tens of thousands or hundreds of thousands of potential sites, we can now look at something like a dozen or something less. And again, that's something that can now be done in a really short period of time. It's really transformative. All of these things coming together.
Right. And now you can also measure some of these things in a different way as well, right? There's new technology that not only are you able to identify more quickly, but the precision with which you can measure some of these things has changed dramatically too.
Absolutely. So that's another cool thing that we've been developing and really the support over this last year. One of the things that we absolutely wanted to do was have the most precise way of measuring DMD. So we want to know when a patient comes in, what level of DMD do they have? And if we correct it, what level do they get to? That's ultimately going to be like basically the therapeutic window.
Yep.
So the most precise way one wanted to determine this would be to count go into a cell and count the molecules. How many DMD messages are there in that cell? Well, we in fact have a way to do that, and the work that the foundation has supported allowed us to get an instrument that we can now measure that. So what we can do, we can take cells either from experiments. It could be from cells in dish or cells from mouse, a large animal model, or patients in the clinic, and measure down to single molecule.
Wow.
How much DMD do they have, and and what the change is.
So this is replacing things like leveraging CRISPR and these types of tools. One of the things as a parent that we've struggled with is, things like biopsies and more traditional methods, which by the way are also highly inaccurate because it's also based on where the sample's coming from, when you're getting it, those kinds of things. So these instruments, just to make sure that, I understand that everybody who's listening does as well, what you're able to do is identify the areas more quickly, and then once identified, measure the efficacy of the treatment, and then you'd be able to take somebody and say, you have DMD or BMD, here's how effectively you are by that, and here's what this treatment will do in terms of efficacy for you.
Exactly. And actually, the next phase that we're going into right now makes that even better. So instead of having to start with the patient and get a biopsy, what we can do is we can take a stem cell and we can now engineer. We were talking about earlier about engineering, like a pig to have a mutation. Well, what we can do is we can take a patient can come in, they can get their mouth swabbed, we can figure out what the sequence of their gene is and what mutation they have. Then we can engineer that into stem cells. Stem cells have the property where you can turn them into other cells. So what we're now starting to do is gear up to do muscle stem cell modeling. So we engineer the mutation into the stem cell, and then we turn that stem cell into cardiomyocytes or skeletal muscle in a dish. And that muscle carries that mutation that we've engineered. So now we tie that together with that instrument, right? We can now measure precisely the molecules of DMD from that mutation leads to. And when we correct it, how much does that improve? What is the level of improvement?
Wow, so you're doing it at an individual basis effectively.
This is really personalized medicine, like coming together as a whole, right? People come in, they have whatever mutation, we treat that mutation, right? Instead of thinking about treating DMD or BMD, it's really treat the mutation, right? It's treat the specific problem.
Yep.
And so that's the plan. The plan is to do that, across the board for really any patient.
Yep.